Welcome to the Eastern Pennsylvania Bank branch of ASM monthly meeting. My name is Vincent Tan. I'm the program director. Last year was our 100th year anniversary, and so now we're stepping into the 101st year. And on behalf of the executive committee, we want to thank all the members and students for your continuing support. Just a quick update, we have recruited Kyle Rubino from Penn and Jason Heindel from University of Sciences into the program committee. And we have a fantastic schedule that for the monthly meeting, we have the speakers and the dates posted on our website. It's also in the meeting announcement emails if you scroll to the bottom. So as always, we want to ask you to join or renew your ASM membership as well as for the branch. If you have already done so, thank you very much. Of course, ASM can advance your science, your career, your network, and foster the community. And we have a new platform called ASM Connect, where you can network with your peers, have discussions in a forum, and attend membership-only events. I know there's some glitch. I think they kind of over-connected recently, but I think they're going to fix it. And lastly, ASM is conducting a survey for your compensation and benefits, and also find out how the impact of COVID-19 has been on employment. So since we're not back in person yet, and we're still using Zoom, I just want to remind you that we're holding all the questions till the end. And so at the end, if you want to ask questions, you can press the participants button and then raise this little blue hand by pressing it. And then I can unmute you and you can ask a question. Alternatively, you can type your questions in the chat box and I can relay the question. So today I have the distinct pleasure of introducing our speakers, Dr. Joris Bell. He's the assistant professor of microbiology and immunology from Drexel University. He received his PhD from ETH Zurich, working with Donald Hilbert, where he did a lot of interesting work looking at the selenium and protein folding. So very much like thiols forming disulfide bonds, diselenide bonds can also promote protein folding, and it's actually more stable than disulfide bonds. And using small molecules, he could restore motility in E. coli in the strain without the SBA. So that strain has a defect in forming disulfide bonds. For his postdoctoral work, he went to UCSD and worked with Mike Burkhardt, where he looked at fatty acid synthase and acyl carrier proteins, where he did some really elegant works by synthesizing the fluorescent probes to look at the process of chain flipping at a single molecule level. Since starting his own lab at Drexel in 2015, he's used mass spectrometry to look at natural products and metabolites produced by bacteria. For example, looking at the presence of vitamin B12 and the different variants produced by bacteria. He's also looked at lipid and fatty acids metabolism. So Joris and I first met at an ASM meeting in New Orleans, way before we came to Philly. And that was a really fun meeting. So hopefully one day we can go back. Personally, I have the pleasure of collaborating with him, and he's a terrific chemist and a scientist. And I think all microbiologists and immunologists need a biochemist on speed dial. And he's a fantastic collaborator and a wonderful friend. So without further ado, thank you for giving your talk. And he's going to tell us about the utilization of environmental fatty acids by bacteria.
Thanks, Vincent. That was a very nice introduction. And yes, I have very fond memories of that New Orleans meeting. All right, let's see. Give me a laser pointer. There we go. All right. So we're all good. All good to go, right? Good.
Yeah.
Perfect. Okay. So yes, my name is Joris Belt. I'm an assistant professor at Drexel University College of Medicine in the micro department. My background is really in chemistry because I'm an organic chemist and a chemical engineer by training. My lab like Vincent said, like we're now 5 years at Drexel. And my lab likes to look at a couple of different projects that also center around the concept of like microbiology and chemical biology of primary and secondary metabolism of bacteria. We like to work with bacteria, but we apply like a lot of chemical techniques to studying metabolism, essentially. And we have projects in the lab that I'm not going to talk about today, really understanding the biochemistry of biosynthesis of secondary metabolites. So how does it actually work on a protein level? And Amanda Platt actually gave a poster talk about this for the ASM before. And other project that we are working on is discovery of natural products. So natural products or secondary metabolites are pretty equivalent terms. And we are looking in like different bugs in the lab, different bacteria that we have isolated from soil or water, etc., where we look for novel natural products. We have, in collaboration, of course, with Dr. Ma, who really started this very big project on cobamide metabolism. So the cobamides are like a family of compounds to which vitamin B12 also belongs. And we have a very big project on that where we closely collaborate. And then last but not least, there's a project here, fatty acid metabolism, where we try to understand how bacteria deal with fatty acids. And that's what I'm going to talk to you about today. In all these projects, we use mass spec. We're very fortunate that we have a very nice mass spec in the lab, and that we can, in all these projects, we can integrate mass spec in one way or another. And in every project of every student, we integrate a component of mass spectrometry, which is highly unusual in a microbiology department, but I think it's to all benefit of all the students to get a bit exposed to this sort of analytical chemistry techniques. I'm going to give a very short introduction about fatty acids. Fatty acids, we all know them well. They're the building blocks of lipids. They influence membrane fluidity. We use them for energy, but of course they are also used for protein post-translational modification. There's a couple of cofactors like lipoic acid, biotin, that are actually synthesized from fatty acids. Many natural products or secondary metabolites have a fatty acid component, so they have a tail somewhere. And some of fatty acids are used for signaling. There are many flavors of fatty acids, from saturated, unsaturated, to like polyunsaturated and branched, cyclopropanes, very long, like this extremely long fatty acid, for example, which we can find in Mycobacterium tuberculosis. There's halogenated ones, where bromines, etc. So there's a very large variety of these fatty acids available in the environment. And that will come clear why that's important. They're synthesized using a very classical fatty acid synthase mechanism, where like the little building blocks starting from like melanyl and acetyl-CoA, we build up in an iterative cycle by 2 carbon units at the same time a fatty acid that at some point gets released from the fatty acid synthase into a free fatty acid. And you can see that during this process, the fatty acids are being carried by an acyl carrier protein. And now I spent a big chunk of my time as a postdoc working on acyl carrier proteins. And we'll get to this little protein that I think is very important in a little bit. Now, in eukaryotes, we have a type 1 system. So we call this a type 1 system because it's one gigantic protein that has like multiple domains. And all those domains are working together to make fatty acids. In this original crystal structure from the Bahn lab at ETH in Zurich, the carrier protein and the thioesterase that are sitting here in the middle somewhere were not resolved because they're so dynamic. In prokaryotes, in bacteria, we don't talk about one protein, but we talk all about separate proteins. So there are a whole bunch of proteins that have to find each other somehow in the cell, and that's what these orange arrows represent, that need protein-protein interactions to find each other in the cell so that they can catalyze the same reaction that happens in eukaryotes in one gigantic protein. Okay. There are many fatty acids in the environment, pretty ubiquitous. Human gut can have actually up to 1 kilogram per liter of fatty acids, whereas ocean water has like typically more in the order of 1 microgram per liter. And in these different environments, we also find a whole bunch of different types of fatty acids in all this in the environment. Sometimes fatty acids are beneficial to bacteria because they can use them, they can degrade them, and they can actually use them for energy. And sometimes they're actually toxic. And I will show you one example where fatty acids are very, very toxic to bacteria. What we are started wondering in this particular project is, I want you to focus your attention on here, what happens in those different environments where you sometimes have very high levels of fatty acids, sometimes relatively low fatty acid content. What happens to those bacteria? What do bacteria do with it? Now, one particular pathway is relatively well known, and that is what we call beta oxidation or fatty acid degradation. It was discovered by Franz Knoop, German scientist, in 1908 in a very, very classical experiment where he fed his dogs aromatic, so fatty acids with an aromatic moiety on it. He fed those to his dogs, then he collected the urine from the dogs and crystallized out of the urine the products. And what he saw was that all the fatty acids that he fed to the dogs were 2 carbon units shorter. So that was the discovery of beta oxidation and how this process works, that fatty acids are being degraded through this fatty acid degradation pathway. And we end up with acetyl-CoA. So it's very well, this is a pretty classic pathway. Now, bacteria can also do something else with fatty acids. They can also incorporate it into other fatty acids, into longer fatty acids through a pathway that we are now calling the AASS pathway, or AS. And I will show in the next slide what this discovery by the Cronin lab, like 20 years ago or so, is interesting, and that I will focus quite some attention on that. There's a route 3, and this is like using the promiscuity of a ketosynthase. So that's the initiation of fatty acid biosynthesis. This enzyme makes mistakes, and it can actually incorporate not only like acetyl-CoA, but it can also incorporate larger units. But in general, we think that this pathway is only limited to shorter chains. So we think about maybe a chain length of C3, C4, C5, C6. These are 3 pretty prominent ways of how bacteria can deal with exogenous fatty acids. Now, during my postdoc, I started this project by doing a very simple experiment where we fed different fatty acids of different chain lengths, going from C1 to C10, to like different organisms. Here we talk about E. coli, Synechocystis is cyanobacterium, Chlamydomonas reinhardti is like a eukaryotic algae, milker algae, and this is a diatom, Thalassosaria pseudonana. And what we did in this case is we fed fatty acids of different chain lengths, and we observe C15 and C17. So say we feed C3 and we find C15 and C17, that means that they actually extended it. Now you can see that we use odd chain fatty acids here, 15 and 17. And we do that on purpose because odd chain fatty acids are very, very rare in nature. If you just have a normal culture of like E. coli, there's very, very little odd chain fatty acids in it because fatty acid biosynthesis gets initiated with like acetic acid, like acetyl-CoA, and then it gets extended. So you will see a lot of C16 and C18 and C14, but you will almost never encounter C15 and C17. Now, if we just start looking at the odd chains and we feed C3, we end up with C15 and C17 and pretty high amounts in E. coli. But you can see it levels off at C5. So we feed C5, but then C7 or C9, we do not see this. So this is very likely a mechanism that involves this promiscuous ketoacyl, the ketoacyl synthase that is the initiation of fatty acid biosynthesis. Now, in the other case, Synechocystis, there's an enzyme called AASS that can actually take up fatty acids and then shunt them into the fatty acid synthase and extend them. And in this case, we see very robust incorporation if we feed C9, C11, C13, and we see a very robust C15, C17, and this is only possible because of this enzyme. And we actually showed that during this project also by making a deletion. Now in algae, it becomes a little bit more complex. We actually don't know necessarily what's going on here, but it looks very much like in algae, it's also a promiscuous ketoacyl synthase. Now, I said already a couple of times EPAASM or S. So this is an enzyme, and the name says it, an acyl carrier protein synthetase. It can take an acyl chain and put it onto an acyl carrier protein. At this point, there are only 6 of these enzymes characterized in different organisms. And what it does is it really takes, this is a carrier protein, it takes like, and we call this the holo form, and I will show you in a second why we call this the holo form, but then it just puts an acyl chain on the terminal tile of the post-translational modification of the carrier protein, the phosphopenetine arm. It's very similar to the idea of a CoA ligase or an acyl CoA ligase, where we have CoA and something is ligated onto CoA. There's very much homology between a CoA ligase and an EPAASM. I told you that I will go over this ACP nomenclature, the acyl carrier protein nomenclature in this field. We have the APO form, which is the naked form. It has a serine residue that is very, very conserved. The serine residue is post-translationally modified with a phosphopenetine arm. This is what we call the holo form. And now the holo form, then we can actually acylate in whatever fashion, maybe through the fatty acid synthase, for example, and we can add like an acyl cargo here. The EPAASM can actually also acylate, and it just puts some other cargo here, for example. Now you see also crypto here. Crypto is when we make an artificial form where we don't have a thiol anymore, but we actually have like an amine at this position. So that is purely artificial. All right. Now, when we started this project, we wanted to look at what can this enzyme, the EPAASM, actually do. We know it can do fatty acids, but can it do more than that? Is there anything that we can use this for? And we tested a whole bunch of different acids, essentially, with different moieties on it, either maybe an alkyne, so we can do click chemistry, or an azide, or something really more exotic. And we tested all these different substrates in an assay where we essentially incubate a holo ACP, so the ACP that has the phosphopenetine arm on it, with the EPAASM and some fatty acid. For example, here on the right is shown incubation with a fatty acid, and we can nicely see then loading of the acyl carrier protein with some cargo. Now, in this case, what we are looking at here is conformationally sensitive urea page gels. Now, this is a bit of a special technique for this acyl carrier protein field that people really like to use. But essentially, this is the urea the concentration of urea is low enough that we are not completely denaturing the carrier protein. So the carrier protein still has a little bit of structure. Now, the holo carrier protein that has the phosphopenetine arm on it is relatively dynamic and floppy. But if we attach some cargo to the phosphopenetine arm of the carrier protein, we get a more rigid and like it tightens up protein, and that will then run like a little bit further on the gel. And that's why, although the mass of the carrier protein plus its cargo is a little bit higher, it will run faster through the gel. So slightly counterintuitive, but important as a technique to look at the loading of these carrier proteins in this field. Now, some of this cargo didn't load at all. The red ones, for example, didn't load at all. All the ones that are in blue and in black actually loaded totally fine. And it gives us a tool now to put like alkynes and azides, which are very unnatural, onto a carrier protein. And we can play this game with like different carrier proteins from different sources. So we can do this with all kinds of different carrier proteins, either from fatty acid biosynthesis, or we can do this with carrier proteins from like polyketide synthesis or non-ribosomal peptide synthesis. And it worked in all these different cases. And we can verify this then with by looking at mass spec, like not only gels, but we actually do mass spec then to actually visualize the loading of the holo carrier protein with, for example, decanoic acid or any other acid that is like shown here, for example. So it's a very efficient tool to install unnatural cargo in vitro onto these carrier proteins. And that's a very useful tool in this field. Now we wanted to take a step further, and we said, well, can we also utilize this in bacteria? So E. coli, if you feed it a brominated fatty acid, it will go in, but then E. coli doesn't do anything with it. But now if we overexpress the Vibrio harvia AASS, which was the first AASS characterized by the Cronin lab, lo and behold, what we look at here is like a strain of E. coli that overexpresses the AASS. We feed it an 11-bromo fatty acid, and we see nicely the formation of C15-bromo this is like a GCMS, so gas chromatography and mass spectrometry trace. And we see nice like that 11-bromo gets extended all the way to C15-bromo, and it's actually quite high levels. And these are the mass spectra that belong to that. Now we brought it a little bit further to algae, and we compared like Chlamydomonas reinhardti, where we don't see this happening. We can feed like the 11-bromo fatty acids to Chlamydomonas, and we don't see anything being extended. But in the case of chlorella, which is a very close neighbor to Chlamydomonas, we actually see nice and robust incorporation and extension of this unusual fatty acid that has like a phenyl ring at the end. So for example, we feed that and we start seeing extension and extension all the way to 14 phenyl C14. So very likely this bug, so this algae actually has an active AASS. Now, one AASS has been characterized in Arabidopsis thaliana, so AAE15, and that is like, this is known. This has been studied like maybe 10 years ago, and we actually repeated these experiments where we supplemented then Arabidopsis in like agar plates with like different fatty acids. In this case, we used brominated fatty acids, and we could nicely see that the wild type of this that contains this gene actually extends it, whereas the mutant, where the deletion mutant did not. So in short, as just sort of the start of the backdrop here, if you feed like propionic acid to all these different organisms, you do get some formation of odd chain fatty acids, right? Propionic acid is C3. So we see like the formation of like C15 in these particular organisms, especially when we overexpress the Vibrio harvia AASS. If we now do this with 11-bromo, we see nothing in Chlamydomonas. We see in chlorella, we see nice incorporation extension, Synechocystis also, and of course also in E. coli overexpressing Vibrio harvia AASS. And this shows like a little bit like that these bugs can actually take up these fatty acids and actually shunt them into their fatty acid biosynthesis and actually go from an 11-bromo to an extended form by utilizing the fatty acid synthase. Where do they go? Well, they don't stay fatty acids by themselves. Bacteria really don't like to have fatty acids themselves around. So what in this particular case, what we did is we looked at E. coli. E. coli ASS acts on the brominated version. ASS puts it on the carrier protein, the Fas extends it, and then it gets put into a lipid. And we do a lipid TLC, so thin layer chromatography, and we look for like, oh, this particular band. Then we look by mass spectrometry in this band, and lo and behold, we see the 14-bromo fatty acid that is like bound to this lipid. And that's, so these brominated lipids actually brominated fatty acids actually end up in lipids. Now, I said that there were 6. Yes, there are 6 ASS enzymes now characterized by both the Cronin lab and the Rock lab, Vibrio, Arabidopsis, Synechococcus, Neisseria, Chlamydia, and L. stipes. The problem is that there are not more than 6 characterized at this point, because they have such a close sequence homology to other adenylating enzymes like acyl-CoA ligases or A domains that it's very, very difficult to actually mine and discover these enzymes. And very often you end up with like something else. We wanted to look in Vibrio cholera as a next step to actually find out if Vibrio cholera has an AASS. And we wanted to do that for two reasons, at least two different reasons. One of them was that the Giles lab showed in 2011 and 2017 that if you feed Vibrio exogenous fatty acids, and especially in this particular case, in these two papers, they used PUFAs. So the DHA and EPA, the very healthy fatty acids that you get at the pharmacy that come from like algae, essentially. If you feed those to a Vibrio cholera, Vibrio will put those into lipids. No mechanism. So we don't know how they actually do it, but if you do feed them, they will go in. So we thought, well, that's a good starting point. Maybe Vibrio cholera has an AASS enzyme that actually takes those PUFAs and actually puts them into lipids eventually. The other thing that is interesting in this field is that fatty acid synthase inhibitors, so that shut down fatty acid biosynthesis, like the molecules cerulenin and triclosan, both of them will inhibit, one of them inhibits the ketoacyl synthase of the fatty acid synthase, triclosan inhibits the enoreductase. They are much less efficient in Vibrio versus E. coli. And people don't know why this particular difference exists. And this is just a comparison between E. coli and Vibrio, but there are many other bacteria where these two antibacterials actually work pretty decently. But Vibrio is a special case where these IC50s are much, much higher for some reason, and some unknown reason, actually, at this point. So if we look in Vibrio and we look for this, and we just look at sequence homology to an AASS, we end up with a list of 10. And that makes that shows nicely what the problem of this field is, that it's in the beginning of this project, we just had 10. We just had 10 gene names, 10 gene numbers and 10 gene names. And we didn't know exactly what they were for. And some of them are predicted to activate short chain fatty acids, some of them succinyl benzoic acid, some of them long chain fatty acids. At this point in time, when we started this, we didn't really know what we're looking for. Now I put here already that this is the giveaway that the VC2484 is an AASS. So we actually in this project identified this particular protein as an acyl-acyl carrier protein synthase. The other piece of the puzzle that we stumbled upon was this, that fatty acid biosynthesis and fatty acid degradation are regulated by FABR and FADR that have been studied by the Cronin lab, involved in the fatty acid synthase and the fatty acid degradation pathways. For some reason, FABR also binds to the promoter of VC2484. And there's no other case where it has been shown that FABR in bacteria binds to an acyl-CoA synthetase or an AASS enzyme, as far as we know. So this already stood out a little bit to us, that why does this repressor actually bind to the promoter of the VC2484? Why would that actually happen? What we try to do next is some bioinformatics. That's the first thing what we thought about, just making like phylogenetic trees where we try to make phylogenetic trees and see if the VC2484 or the VCAASS grouped with anything else. And it made a pretty unique separate clade. Then we did protein networks. This is a pretty similar idea that we're grouping proteins based on sequence together. And what we can see here is that the VCAASS actually grouped together pretty closely to the Arabidopsis enzyme and to the Synechococcus and Synechocystis enzymes. So they all grouped together here, where all the other enzymes that I showed you this giant table of formed like separate groupings that did not group with any of the other AAS enzymes that have been characterized, like for example, the Vibrio harvii AASS. So it gave us a bit of a feeling that perhaps this enzyme, this VC2484 or VCAASS, is actually an AASS enzyme. What we did next is also some more bioinformatics, but in this case, we did structural modeling and docking. At this point, there is no structure of an AASS enzyme. We are actually working on it, but there is no structure. So what we did is we constructed models, and then we docked carrier proteins to the models. And lo and behold, when we did this with the VC2484 or the VCASS, all the carrier proteins, and there's like 10 different models here, all the carrier proteins dock to one particular site. And this suggested that we have a model of convergent docking, that perhaps there is a site here that actually interacts between the ACP and the ASS. If you do the same experiment now with FAT-D, which is the acyl-CoA ligase or acyl-CoA synthetase that starts the fatty acid degradation, which has homology because it's in the same class of enzymes, you get this carrier protein that docks all over the place. There's all this spaghetti or all the 10 different models, and there's no like particular location that this likes to dock to. And it makes sense because FAT-D acts on CoA and not on ACP. All that bioinformatics is nice, but in the end, I have to go to the lab. We did a simple experiment where we took a Vibrio, we fed it like a odd chain fatty acid that you normally don't see, C13. And we did this in like a whole bunch of different transposal mutants in these particular genes. And we looked for, can we find C15 or C17 in any of these, right? If we make a deletion or we make a transposer mutant in one of these and we can still see the product, then likely that is not an ASS enzyme that is active in this pathway. We did this, and then we found that the VC2484, we can still see C13. That's what we fed. That goes into the bacteria, but it cannot be extended. And we don't see C15 and C17. We have some other cases where we don't see C17, and we don't, at this point in time, we don't actually know why that happened. And that's something that we're working on. Now, then, of course, we have to do in vitro work. We had to show that actually in vitro, this enzyme can load fatty acids onto a carrier protein. And we did that again with our conformationally sensitive urea page. Here on the top, we use Vibrio harvii AASS. This is the enzyme that was the first ASS. It's the one that is well characterized. And we can see nicely on the right side here, loading of like 4, 10, 11, 14, and even C16 fatty acids onto the carrier protein. And these are all acyl ACPs. This is well known and forms like a nice little ladder essentially. Now the Vibrio cholera AASS, it also loaded, but there are some, we can still see holo ACP here, right? We don't see any of the holo ACP, that's the starting material. We don't see any of the holo ACP anymore here, but we do see bands here for the acyl ACPs. It means that this is just it's just a worse enzyme. It's just not much less active than the Vibrio harvii AASS enzyme. And then we can, of course, in my lab, we can then do mass spec, where we actually can show that we go from an holo ACP species to a C11 loaded ACP species. We can do mass spectra, we can deconvolute that and nicely show that we go from an holo mass to like a mass of C11, for example. But you can also nicely see that there is still the mass of the holo form in there, and that suggests that the Vibrio cholera AASS is just not very active. I told you that the AASSs and acyl-CoA synthetases or acyl-CoA ligases are similar enzymes. And indeed, many of the AASS enzymes will also activate CoA. That makes it a little bit complex, of course. Here on the right is some data that we have collected where the TTHA 0604 is a very well-known acyl, like a long chain fatty acid acyl-CoA synthetase from Thermus thermophilus. So this one we set to 100% activity. That's the most well-studied, one of the most well-studied CoA ligases. And then we compared that with the activity by using mass spec, but also by doing some UV vis photo like assays. We compared then the activity of the Vibrio harvii, the Synechocystis, the Arabidopsis, and the Vibrio cholera ASS with this known acyl CoA synthetase. And you can see from this that although the Vibrio harvii, so the Vibrio harvii, the Synechocystis, and the Arabidopsis are all known AASSs, so they all activate ACP, they also all activate CoA to a different extent. And our newly identified Vibrio cholera ASS does the same. And we show that, of course, by mass spec here on the left. Now, the picture in Vibrio cholera is like this now. We have, Vibrio has a unique lipase on the outside where it can actually degrade lipids on the outside. So it can actually make fatty acids on the outside of the cell. Then these get imported into the cell by using an importer or transporter VAD-L. Vibrio is unique that it has 3 of them. So it has VAD-L 1, 2, 3. This is not seen in other bacteria. And then the fatty acid is in the cell, and then we can actually maybe go through a CoA ligase and make CoA forms. We can also go through an AASS step here and then make our acyl ACP species. And of course, these can then be further incorporated into lipids, or they can be extended through the fatty acid synthase and then again incorporated into lipids. And this is our current like sort of working model of how this works and how maybe Vibrio can actually circumvent fatty acid biosynthesis inhibition, because you can imagine that Vibrio can take fatty acids from the outside. It can actually then use those fatty acids, maybe via an AASS mechanism or maybe via a CoA ligase mechanism, make lipids out of them, but it does not involve the fatty acid synthase anymore. It just uses the environmental fatty acids. Now that makes it perhaps interesting to start a project where we inhibit the ASS, right? Because if the ASS is important for this process, in the case that the fatty acid synthase is inhibited, and it really starts using environmental fatty acids, maybe inhibiting the AASS, it becomes an interesting project. So we started the project with Dr. Yaremko at Hofstra University, and we actually published this paper like last year, where we synthesized a new inhibitor of an AASS. And you can, if you're a chemist, it essentially what it does is it mimics the acyl AMP intermediate of like this enzyme. So this is normally formed in the active site of the enzyme. And normally there's a phosphodiester bond. Now we replaced it with a sulfonyl bond here. And this bond is like stable, so it cannot be broken anymore. So this molecule sits in the active site of the ASS enzyme and then inhibits it. And we tested that, of course, using our conformationally sensitive gels again. And again, we can see here that if we incubate now the E. coli ACP with the Vibrio or the Vibrio cholera ASS, we can go from an acyl species that is normally formed, we add the inhibitor, and we only have holo, and we don't have the acyl ACP species formed anymore. And the same for the Vibrio, and the same for the Vibrio cholera ACP. So now we have an inhibitor in hand that in vitro at least can inhibit the formation of like these acyl ACPs. And of course we confirmed this by doing mass spec. And then of course we wanted to test it. So what does it do to bacteria? So we fed this inhibitor to Vibrio cholera and nothing happened. And we were not surprised by this, because in like these conditions, these inhibitors don't do anything. They maybe inhibit something, but the bugs are not dependent on the fatty acid uptake. So in this case, these inhibitors don't do anything. Now, when we start now using a very strong fatty acid synthase inhibitor, cerulenin, we can actually shut down growth of Vibrio cholera. It has been shown. So we can shut down growth. And we can rescue this by adding fatty acids back in. So we can actually, in this case, we have cerulenin plus fatty acids. So we can actually rescue inhibition and cerulenin hits the fatty acid synthase. Now, if we make it a combination of cerulenin, fatty acids, and these new inhibitors of AASS, we can go from like the line over here, the growth curve, to the growth curve here in the bottom, where we actually see inhibition happening again. So now we essentially switch the bug to like dependency on fatty acid uptake and fatty acid utilization from the media. And if you then give it like the AASS inhibitor, you end up with this profile. Yeah. We also did this in a little bit of a different context. In a way, we used in this case isoniazid, which is not which is also an inhibitor of the fatty acid synthase, but has never been used in Vibrio cholera. And we did this in a different media, so that's why these growth curves look pretty different. But in the case of isoniazid, you can actually switch. You just kill them essentially. The growth is like completely inhibited. You can rescue it again with fatty acids, and you can rescue it again with fatty acids here in the middle curve here. And if we do the same experiment now in a deletion of the Vibrio cholera AASS, we don't see that rescue anymore. This is pretty preliminary data, so we're still working on this particular portion of the project. Now, we also did this in a slightly different setup, and this is in we actually let Vibrio cholera grow on like M9 media supplemented with different fatty acids. And what we saw in this case is that when we do that, Vibrio grows fine when there is like short chain fatty acids present or long chain fatty acids present. But Vibrio refuses to grow when you give it decanoic acid, C10. We're not the first ones actually seeing this. The Cronin lab actually saw this before, that if you give C10 to Vibrio cholera, you inhibit growth. And when we made our VCASS knockout, we actually saw that that particular strain is growing. So this is another indication that perhaps the VCAASS is really important in this process of metabolizing exogenous fatty acids further down the line. Now, why C10 is toxic and what actually happens here, we actually don't know. And this is still a project that is ongoing at this point. So I talked really fast, but now we have a little bit of time for questions. What I hope to show you now is that VC2484 is an acyl carrier, like an acyl carrier protein synthetase. So we have identified a VCAASS because it does load fatty acids onto an acyl carrier protein, whereas if you do the same experiment with an acyl CoA synthetase, that does not load fatty acids onto an acyl carrier protein. And we have tested that many times. The AASS allows some bacteria to circumvent Fas inhibition. And this is pretty important because there are at the moment at least 2 drugs in clinical trials for Fas inhibition. So we think that maybe this class of enzymes can help these bacteria circumventing Fas inhibition. We have synthesized with Dr. Yaremko, we have synthesized some inhibitors that we can now use as lead compounds to inhibit AASS. But of course, this goes then together, right? We have a Fas inhibition and AASS inhibition at the same time. What is very important for this step is that we need a structure. So that's what we're really working hard on, getting a structure of an AASS enzyme. And when we published this originally a long time ago in 2015, it got picked up by the journal and they wrote like a little commentary on that with this punchy line of ACP-AAS, you like it, because they thought it was like the ASS is like a Swiss Army knife because we can use it to install all kinds of different cargo onto these carrier proteins. So with that, for this particular project, I really would like to thank Amy Ma, who made like lots of contributions to this project, including like knockouts and etc. Amanda did a whole bunch of like these gels that I showed, the conformationally sensitive gels. Tyra and Scarlett were two master students in the lab that really worked hard on growth curves, and Gabrielle too. Some of the data that I've shown you came from like the Burkhardt lab, the Hildebrand lab, and Noel lab when I was doing a postdoc there in San Diego. And these projects were funded by the DOE. Right now, we're collaborating very closely with the URMCA lab on these inhibitors, but also with the Gulian and Zhu lab at UPenn, where we got some strains from, for example. I should not forget to thank the Cure. So this is funding from the Commonwealth of Pennsylvania that supported this project with money. And with that, I'm happy to take any questions.
Thank you for a very interesting talk. We have the first question.
Oops.
I think you're unmuted.
Okay, hi. Hey, Joris. Great
Hi, Gil.
Yeah, I should ask you this before, but so the question I have is about something very naive, so please forgive me. So fatty acids are generally not soluble, right? So so how are these actually taken up, and in which form are they taken up? So are they bound to some other stuff, you know, like proteins or other things, and then they're taken up and then they're taken apart? How does that work?
Ooh, that's an excellent question. So on one hand, people actually don't know necessarily how this is being taken up. People in the lab have always used solubilization using detergents.
Right.
So, but that is not, of course, not what happens in nature, very likely. So we actually don't necessarily know how this process works. And I mean, the discovery of that full A lipase on the outside of Vibrio cholera, what essentially just grabs lipids out of the solution and then cleaves the fatty acids from the lipids, and then the fat L is right next to it, and the transporter essentially transports then fatty acids directly into the cell. That is the model right now for Vibrio. That is not universal because there are very few bacteria that have a lipase on the outside.
No, but the lipids also need to be bound to something, right? Because they're not soluble either. So they too need to be carried in some way. So I mean, it's the whole, you know, hydrophobicity versus the water, you know, hydrophilicity that the bacteria sort of live in. And I'm just wondering how that works. So,
yeah,
it's a similar problem for Plasmodium as well. So I mean, so and we do have to make them into, you know, combine them with proteins in order for them to be taken up properly, like with the BSA and things like that. So.
Yeah, that's a good point. I have to say I've never looked into that, but that's a good one. We should actually have a look at that. Yeah. Thank you.
Okay, so I have a couple of questions. So you mentioned that, you know, there's the fatty acids very low abundance in the ocean, right? So that's Vibrio and Javier. And that makes sense if they want to incorporate that if they encounter it as a nutrient supplement. But of course, chloride causes disease in the human, and then she said there's a much higher abundance. And even they can incorporate DHA and EPA, and as we try to understand how, you know, even the downstream lipids are affecting, how are they? So first question is, are they expressed during infection? And second, how would you think if they do express it when they encounter all these host lipids, how would that affect, you know, the membrane fluidity, the pathogenesis, or even the shape of the bacteria, right? So
yeah. Those are some very excellent questions that I have to say that I don't know, because it's just at this point it's just not known. I mean, the one thing, so in the ocean, for example, low concentrations of fatty acids, there's quite a bit of DHA and EPA in the ocean, but it's at low concentrations. The other lab, the Giles lab, showed that these fatty acids are being incorporated into lipids. So they are not using those necessarily as an energy source while they're in the ocean. They actually incorporate them into the lipids. The question is, why do they do that? That is pure speculation at this point. There's no great data on that necessarily. What actually?
And that seems to may have an even bigger effect since they're unsaturated, right? So
yes,
the kinks might affect the
totally.
The
right?
downstream.
It will change membrane fluidity. It might change shape or etc. In the host, the problem is also interesting because there you're exposed to very high concentrations. So there is so much fatty acid around, and we can do these experiments in the lab where you give them those high amounts of fatty acids in the media, and they go nuts about these things because they actually start putting some of the exogenous fatty acids that they normally don't make themselves. They can actually start putting them in their own lipids. What that does to the bug itself, that was not within the scope of this particular project yet. We would have to do some careful microscopy, etc. We haven't done that. Whether this is activated during infection, we don't necessarily know. There are some good like RNA-seq papers essentially, or reporter papers where they have done like the whole library of all the different genes in Vibrio under different conditions when you put it into a rabbit or when you go from media to an environmental environment. We don't see that much happening necessarily on these fatty acid synthesis or fatty acid degradation genes at this point. So I don't know the answer, but that's of course, this is bugging us too.
Yeah, that's very interesting. Jason has a question.
Hey, nice talk.
Hi.
Thanks for entertaining us and teaching us some stuff. Totally not my field, so I'm going to ask maybe a simple question. Your cholera enzyme looks in the data looks to have low efficiency relative to the Harvey I enzymes. Is that just because of the purification protocol, or do you think there's some other post-translational modification you need to do, or is it a substrate that's not being provided appropriate substrate for it?
Yeah, that's a good question. We don't think it's post-translational modification necessarily. The purification, when we purify this, we run the Vibrio harvii and the Vibrio cholera at the same time, more or less, and they purify and express more or less the same. There's not a huge difference between these. We think it has more to do with substrate. We don't have the best evidence for it yet, but normally what we do with these kind of enzymes is we run a whole panel of different substrates, ranging from short chain fatty acids all the way up to like very long chain fatty acids. And we can definitely see a big difference in how the Vibrio cholera enzymes responds to these fatty acids. And it seems that the Vibrio cholera likes a little bit shorter ones much better than the Vibrio harvii. Now the next question, of course, is why? And we have no clue why that would be necessarily. But this is not so uncommon, because if you look at the Arabidopsis or the cyanobacterial ASS enzymes, they also have they all have different substrate specificities. So the Vibrio harvii was the first one characterized, and that really likes chain lengths around 14. But the other ones much more prefer like shorter or even longer. So, but yeah, that's a good question. Yeah.
If they knock it out in Arabidopsis, how does that change the plant physiology?
It does not change the plant physiology. So the mutant does look the same. Now, what we then started to do is mess around with it a little bit, and we actually put like different fatty acids. So Arabidopsis we can grow it on agar plates. So you can actually, in the agar itself, you can pour in like different fatty acids. So we started pouring in the agar plates different fatty acids, brominated ones, long chain, short chain. and you start changing physiology a lot, like really weird things with like big leaves or like short stems or no roots, lots of roots. We've never published it because this was, I don't know, we didn't know how to interpret it necessarily.
Yeah.
Yeah. But it, yeah, I think for Arabidopsis people, that's kind of interesting.
I'm going to jump in with another question. The decanoic acid toxicity or lack of growth, do you think that's some kind of anti-quorum sensing, some signaling, or is it actually disrupting the cell physiology in some way?
Yeah, we've put proposals in to look at this, but never got funded. But so this is also, so the C10 toxicity, so the decanoic acid toxicity is not unique to Vibrio. This also happens in other bacteria. This even happens in some eukaryotes. And so we don't necessarily know what the deal is there. We assume that it since in the AASS deletion strain, we don't see the toxicity, we assume that the decanoic acid needs to be loaded onto the carrier protein to become toxic. So something gets clogged up, and or something goes wrong. So maybe the C10, so you make the C10 ACP, that becomes then a substrate for lipid biosynthesis. It can also be perhaps a substrate in the direction of lipoic acid or biotin. And either it goes wrong in the cofactor arm or it goes wrong in the lipid arm, where you end up with lipids with very short fatty acids, which is not good for most organisms. We would like to know the answer to that. And we still have some plans to address that at some point.
Oh, cool. Very interesting.
Yeah.
Okay. Actually, perfect timing. So thank you again for a wonderful talk. We'll see each other, hopefully in person soon enough.
Definitely. Yeah, thanks for the invitation. Very nice to you all.
Yeah. Thank you very much. And we'll see you guys next month. All right. Thanks.
Okay.